Modern biology generates data far faster than any human can analyze it. A single study can easily produce billions of data points. To make sense of this, a biologist must piece together dozens of specialized software programs, write custom code, and manually format databases. Human bandwidth simply cannot keep up with this fractured process, leaving vast amounts of valuable experimental data unexamined. Imagine giving a system a plain English instruction, like analyze this genetic sequence, and having it autonomously select the right tools, write the code, and run the experiment for you. That is the premise behind Biomni. It is a general-purpose biomedical artificial intelligence agent built specifically to execute these complex, multi-step research tasks on its own. By offloading the rote programming and data parsing, researchers can step back from manual labor and return to framing the actual scientific questions. To give the agent a working environment, its creators built Biomni E1. They designed an action discovery AI to read 2,500 recent biomedical research papers and extract the exact tasks, tools, and databases scientists rely on. This mapping created a unified workspace containing 150 specialized biological tools, 105 software packages, and 59 distinct databases spanning 25 areas of biology. Instead of relying on rigid templates, Biomni uses executable code to interact with this environment. It reads a user's prompt, plans the steps, and dynamically writes the scripts needed to link these tools together. To test this architecture, researchers handed Biomni a massive dataset, 1.4 billion raw heart rate and step measurements from over 1,000 Fitbit users. This chart shows a disrupted circadian rhythm biomarker extracted from that data. Working entirely on its own, Biomni wrote an analysis pipeline to process the raw readings and correctly identified 6 validated early warning signs of COVID-19 infection. Looking at these findings, Biomni successfully reconstructed the population's risk scores and biomarker correlations. In similar diagnostic tests across different medical fields, Biomni matched the accuracy of human specialists, but reduced the time to completion from hours to roughly 3 minutes. This speed removes the manual bottlenecks that traditionally leave massive longitudinal datasets unexamined. Identifying targets on a computer is only half the battle. Those digital insights have to be translated into physical wet lab experiments. Researchers asked Biomni to design a molecular cloning protocol to insert a specific CRISPR guide RNA, a targeted genetic edit, into a cellular construct. The agent parsed the plasmid structure, designed the required genetic sequences, and output a complete step-by-step laboratory instruction manual that anticipated the exact edge cases a senior geneticist would. When human bench scientists executed Biomni's protocol exactly as written, it worked on the first try. Sequencing confirmed genetic edits inside the resulting bacterial colonies. The final step is removing the human hands altogether. To do this, developers integrated Biomni with PyLab Robot, an open-source framework for controlling automated laboratory machinery. This code shows Biomni configuring the exact layout of a robotic deck. Given a simple text command to perform a liquid transfer, the agent generates production-ready Python syntax that handles tip selection, serial dilutions, and volume validation. This image shows the direct result: a Hamilton Star liquid-handling robot executing a multi-step drug dose response experiment based entirely on Biomni's code. The agent has crossed the threshold from digital analysis into physical lab operation. Biomni's path from a digital text query to physical lab execution marks a transition in how we bridge research domains. Weeks of tedious manual coordination, software troubleshooting, and literature review are condensed into an autonomous pipeline. However, an agent capable of generating accurate biological protocols on demand carries serious risks. The same system that accelerates disease research could lower the barrier to creating dangerous pathogens. This dual-use reality requires strict policy frameworks. The developers of Biomni emphasize the absolute necessity of responsible, transparent development to ensure these tools maximize societal benefit while locking out bad actors. Ultimately, Biomni is designed as a tireless collaborator. It handles the rote mechanics of the scientific method, not the inspiration behind it. By absorbing the logistical burden of running experiments, these agents allow human researchers to focus on the creative leaps and conceptual breakthroughs that machines simply cannot make.